You had a crazy viral moment last week when your biological computers were playing Doom.
Yeah, you can say that. And it's actually quite interesting because the Doom story is almost 2 weeks old. And there was one uptick of interest, and that was when Elon tweeted about it. And then on Sunday, it somehow got like a second wind of virality, and that one was through the roof. Millions of views, multiple channels. I mean, the really annoying thing is now I have all these crypto bros coming up to me and say, hey, are you going to claim your Solana? And I'm like, bro, I run a legit business here. I can't just take Solana.
The real problem, I feel like every time there's some kind of innovation, there's a bunch of crypto spam that comes with it.
Oh my God. And the problem is I like crypto, and I think there are some really cool approaches to it. And I'm trying to advise the community. I'm like, guys, I don't care about creative fees and all that stuff. Go set up a DAO, put money in, vote amongst yourselves to fund projects kind of thing, right? Not just give it to a creator, because that's how you get rug pulled, right? You need a proper constitution. You need a way of like distributing money and so forth. I think this is how we can actually change the paradigm of, hey, I'm a researcher here. I need to go spend time writing a huge grant process that is going to be like probably denied anyway. Why don't I just go to the crypto community and pitch my idea, and maybe I can get some funding that way?
Yeah, and the crypto community seems to invest and spend money in a very different way. It's like the money came very fast.
Yes.
For some of them.
Yep.
Came in huge quantities, like there's hundreds of billions of dollars sitting on the balance sheet of, frankly, quite odd people.
Yes. And I mean, it's incredible as well. I think, what was it? Almost a year and a half, 2 years ago? the Tether Foundation put $200 million into BlackRock Neurotech, which is like one of the oldest and probably not the most exciting BCI company, but I don't know, they must have seen something in that.
Must have. So why don't you explain the device?
Yes.
How does the actual computer work? What's going on?
Yes. So what we have here is the CL1. It's the basic compute unit of the Cortical cloud. And let's go one step lower here. So think of it like Matryoshka dolls, right? There are like little dolls that are nested into one of them. The smallest component is actually this. I'm going to show you, and you can handle it and maybe show your audience. It's a little bit cracked.
Doesn't have any live neurons in it, does it?
No, but we'll see some in the lab later today. This is a glass chip called a microelectrode array. And what it has are 64 electrodes down in the center portion that radiate to the external bits. We grow stem cell derived neurons into that chip.
Human stem cell derived neurons.
Correct. Human stem cell derived neurons. And the neurons mature over time, and within about 2 to 3 weeks have electroactivity. Because they're electrically active and they're sitting on a conductive material that radiates the external bits, we can make contact with that using our device here to read that activity. And we can also provide a bit of electrical stimulus back as well. So in that sense, we have read-write access into these biological neurons. So that's the smallest, basic compute unit.
Let's go a bit deeper into this. So where are the neurons and how many are there? And are they self-organizing?
Yeah.
Explain how it works.
So as you can see, it's kind of like a petri dish, right? And so the petri dish design is
inside there.
Yeah. So you have the well, and the well is to hold cell culture media that keeps the cells alive, allows them to absorb glucose, respirate, have waste produced and so forth. At the base of that, if you look really closely, you'll see tiny electrodes at the base that terminate in the center, in the origin, and we can grow.
These are these lines that are coming out into the middle.
Exactly, yeah, and we'll see some of that under a microscope in the lab. You can grow 200,000, which is what we usually do, to 800,000 to millions of neurons on an organoid, right? So the scale is actually quite large, and we only take up a tiny portion. So if you can see, there's a tiny little blob there, right, where we have what we call a microfluidic PDMS structure. So if you look down really closely, these are very small wells where we can very finely place neurons in them, and they have these tiny little microfluidic channels that we can use to control the growth of the network. So in the previous work that we did, we kind of let the cells grow wild and tried to shape them using information. That works, but we found that if you also give them a little bit more structure and you could actually ablate channels or add new channels in, you have a lot more control over the network and you get a bit more compute from that. So it's still very active research that we're doing internally. We haven't really rolled it out to our cloud users yet, but it's something that is probably in the pipeline.
Right. And if you have 800,000 neurons, is there a way of converting that into parameters like you'd see on a large language model? Like, is it
Um,
800,000, presumably it's all the connections between them. There would be many more connections
well,
than neurons.
not quite. I mean, it's interesting because if you think about how we think of large language models, they seem to get better with more scale. I don't think biology really works that way. For instance, right, like a chihuahua isn't that much dumber than a Great Dane or something like that. Even though the skull size is much smaller, they have the same number of neurons, probably same number of synaptic connections. I think it's more about the processing of information, the embodiment of it, that actually determines how good a particular organism is at processing information. For instance, there are many organisms that have more neurons and bigger brains than us, yet we are smarter than them, or at least I think we are. So, an elephant, for example, they're pretty smart, but I think humans are still much better at most tasks than an
But there's a lot of
elephant.
selective pressure on this, right? Like brains are very energy intensive. So why do you think that would be the case? You know, if there's that much pressure on keeping brains as efficient as, why do you think it would be the case that, you know, an elephant is that large? Is it because their actual nervous system is so large because they're a bigger animal?
Probably. I mean, it is, they're a larger animal, so, they have a larger brain. Same thing with a whale. But it's, the evolutionary pressures on them, right, did not force the level of connectivity and the complexity that their brains have evolved as opposed to Homo sapiens. Because if you think about it, we're pretty bad at most things. We're the weakest ape, the one with no hair. We're pretty bad at most things. And the only reason why we've succeeded as a species is, the brain, right? We use our social ability to form hunting groups. We develop clothing so that we can colonize the regions of the planet, and the ability to make tools and augment our abilities is, I think, the reason why we have been so successful as a species.
Well, I guess it's really the size of the network, right? You know, if you have
Yes.
the size of the United States
Hmm.
that can organize and support advanced technologies that even the smartest tribe of 10 people wouldn't be able to do.
I think it's that organization.
Hmm.
Yeah. So, go back to this chip here. So we grow about 200,000 neurons, which is less than what we started out with. We started with 800,000 in the initial Pong paper, but that's because at that time we had no idea whether you could get it to work. And so with little funding and little time, you just throw everything at the kitchen sink and see what happens, right? So we started with a lot of neurons and prayed that there was an effect. Once we saw that effect, we started to go the opposite direction. Rather than growing larger systems, we wanted to go smaller. And that's for two reasons. A, if you grow smaller, you have less complexity, and therefore you understand what is actually happening at a more fundamental layer. And the second point here as well is, from a business perspective, the smaller the system you get, the more replicable you can make it, the less costs involved. So it's a balance between, performance versus, how much cost is involved in time.
So going back to that Pong example, so that experiment, you had a sheet of neurons, you were shining light on it, if I remember correctly.
No, same technology. Electrical, yep.
Electrical, and then they were self-organizing and basically outputting something that you were reading.
Correct.
And then how did you go from that step to teaching them how to play Pong or getting them to play Pong?
So in the Pong experiment, we encoded the XY position of the ball using a mixture of rate and place encoding. We just said, hey, you neurons in this upper region, you're the sensory neurons. Some of you are going to be corresponding to positions on the y-axis, and we're going to strobe the distance of your paddle to the ball in terms of frequency. So the faster we signal to you, the closer the ball is getting, the further away, the longer the period. So that was very basic on how we did that. The Doom is actually a little bit more interesting. So we didn't actually do the Doom experiment. To be completely transparent, this is actually a project and run by a computer science student by the name of Sean Cole from the University of Sussex, which is actually far more interesting, right? Because we didn't do it, but he used our system and our tools to actually develop it, which has, I think, been very fulfilling because now we actually get to see the hypothesis, the thesis that we had from the beginning, where if you build the tools, the people will come, which was always the big question. A lot of investors have asked me, are you sure that people will come even if you built it? I'm like, I think hopefully they will, right?
So how do you go from that Pong example to multiple of these to then being online and playing Doom?
Yeah. So we go back again in time. So we started in 2019. We got funded to really see if we could actually get these neurons to do something. We proved that was possible with that paper in 2022 that came out with the Pong. And then from there on, we had a lot of inbounds asking us two things. When are you going to do Doom when, Doom next kind of thing? And then the thing that really triggered our thinking about where we were going to go next were a lot of questions from my academic colleagues about, which machine did we buy? What software did we write? What cells did we use? And, being a second-time founder, when you have questions about where do you buy something, how much does it cost, what software do you write, and you have it multiple times, you start thinking, is there actually a market here? And so, talking to our academic research colleagues, it kind of dawned on me that one of the reasons why this space has not really taken off is that it is kind of like a cottage industry. Now, if you think about a cottage industry, it's very inefficient because every village has its own shoemaker. And so you don't get economies of scale. You get different shapes, different sizes that don't quite match. So it's very bespoke. And you go at a much slower rate that way. And a lot of these labs would raise funding from their grants program, let's say 5 million or so. But out of the 5 million, they probably spend at least 1.5, 2 million just developing the hardware to do the experiments or write the software. And they're good, but they're not experts at developing hardware or making it at scale. And if you have 5 or 10 other labs doing the same thing, then now you've wasted, say, $5 million with duplicated efforts. So when I thought deeply about that, I realized, what we could do to help contribute to this space was let's take on the onus of building the hardware, building the software, and doing the protocols for the wetware, package it up, and offer it to the research community so that they can do research instead of reinventing the wheel over and over again.
Yeah. And in terms of getting actual useful compute out of these cells, how does it work? Is there a reward function? What's actually going on?
Yeah, there's a reward function. There are multiple different approaches to it, right? I personally like reinforcement learning because, I'm from an ML background, but there are people looking at it using reservoir computing, right? Which is more of a training technique that is, unsupervised, that is clustering kind of stuff. I think it works as well, but I'm less of a fan of that approach. But the way you would actually train these things to get any compute is to let the neurons do what they're good at, which is to be entropy reducing machines. And you give them a program, in this case, the game of Pong, Doom. So you can actually give structured information back about rules of the game. So in the case of Pong, you have a ball that bounces against the 3 walls, and if it hits the back wall, it's game over. And if it hits the paddle, it keeps the game going, and so on and so forth, right? These are all rules that gives structure to the information that we give to neurons. Once you have that, you just let the neurons essentially fit to the program, right? Where you steer them in a particular direction by giving them a reward or a punishment signal kind of thing. So in reinforcement learning, you would do that. And, we see this happening with...
What are those reward and punishment signals you're giving them? Are you giving them more glucose? Are you
no, we're
poking them with a stick? What do you...
No, we vary the amount of information entropy. So the predictability of the signal we saw had an effect where you could actually push and pull them in one direction or another.
So they want to be organized. Correct. They want to self-organize, and the longer the game goes...
Yeah. So essentially so this is from a theory developed by Carl Friston at UCL, right? Which is known as the free energy principle. On a very high level, what it actually talks about is that if you have a system like a brain, neurons, or organism or whatever, it has its own internal distributions, right? The external world has its own distributions. And the whole point of what these biological systems are trying to do is to minimize the amount of information surprise, i.e. the internal prediction delta versus the external world. If that is what's driving them, we realize that if we could change the environment so you could make it more or less predictable, you could actually force the neurons to conform to one path or another.
It's interesting that it seems like that's a simple basis for what's going on in the networks of neurons. They're really just trying to make sense of the world around them, and that in itself is enough of a reward.
Well, I mean, it is a survival function, right? If you think about it. Because the better you can predict the external world, the better you can respond to threats. If you can predict ahead of time that there's an eagle coming down to you and end your life, you'd run away quicker and therefore you survive and pass on your genes. Conversely, if you're an eagle and you can see a rabbit and you can better predict where the rabbit's going to go, then you get your lunch or whatever and you survive the next day and you reproduce. So information prediction is actually a survival mechanism for organisms.
do?
Well, I mean, in most cases, we are doing that, right, in our heads. For instance, optical illusions, right? So the best one, the easiest one you can just do is close one eye and look straight ahead. You'll see your nose. Close the other eye. You see your nose. You open both eyes. Your nose is like deleted. And that's because the brain is not simply processing information, it's actually generating a representation of the world. Hence, why people like Jan LeCun and so forth are talking about world models, right? Because that's what we are doing so efficiently in our heads, right? Building world models, constructing the three-dimensional space that we operate and making sense of the photons that are reaching our eyes.
It's fantastic. It's really interesting science. And then to turn that into a product where you're actually putting in inputs and getting in outputs.
Yeah, certainly. I mean, there's still a lot we don't know about how these systems work, and we are just at the cusp of like delving deeper in understanding.
Is that enough? Like, do you think there's more to it, or is that simple principle enough to explain most of what's going on?
I mean, I think there is a lot more happening, and in most science, all we have are abstractions, right? And we latch on to the best abstraction that we have at the time to explain the phenomena that we're observing. By all means, I think that the affinity principle is not the end point. There are probably a lot of other ways of explaining this. And it also has its flaws, for instance, right? Like there's this thing known as the darkroom problem, right? Which is that if all these systems are trying to minimize their information surprise, hypothetically speaking, here's a thought experiment. If you put somebody or a neuron or brain or whatever into a dark room, why would they ever leave the dark room? Because now your information surprise is zero, right? Everything is entirely predictable. But yet we know you won't stick in the dark room, you will actually leave it, even though that means increasing your information entropy. Sorry, your information surprise. So it doesn't answer everything. I think this is a field that is ripe to do more experiments, to come up with more theories that better explain the phenomena that we're observing.
I feel like there's a big difference between science and engineering. You almost need to, I wouldn't say understand things better, but you certainly need to understand them extremely well and be able to manipulate them and get them to do things that you want them to do.
For sure.
What are the main things you've learned about networks of neurons through this process, through turning them into a computer that can be accessed?
Yeah, so internally what we've learned is that there are a whole bunch of very deep science around this. So one of the first things that we learned that was quite interesting and surprising is this concept known as criticality. There is the similar concept of criticality in physics, right? In physical systems. So let's say water has two critical points, right? It's the phase change between being a liquid to a solid and a liquid to a gas. So at 0 degrees Celsius and at 100 degrees Celsius, something really interesting happens is that 50% of your water molecules can become a solid, or it can turn into a gas, or vice versa. Neurons actually have a very similar property as well called neurocriticality. And it's really interesting because most healthy neurons sit at that critical point of what we call a branching ratio of 1. There are many other measures of criticality, but branching ratio is probably easiest to talk about. And why having a critical point of 1 is really important is because it helps propagate a signal all the way to the end, but it doesn't overwhelm the network, right? So if you think about a branching ratio of 1 means that one signal from a neuron will continue passing on to the next one and another one and so forth. If you have less than 1, like let's say 0.8, there's a 20% chance that the signal will not propagate to the next network. So you end up with a very depressed state and the network doesn't completely propagate the information through. On the other hand, if you have a greater than 1 branching ratio, where there's like a 20% chance that your one signal will now branch into 2 different neurons, right? And over time, you will compound on that. You end up with an entire network that gets turned on, essentially epilepsy, right? Seizures are overactive networks. And so what's super interesting is that when we did our experiments with Pong, we furthered that by looking at the network dynamics. This actually is a publication in Nature Communications, I think in 24. We found that when neurons are not put into a game world where they don't get that structured information back and forth, they fall out of this criticality zone. So they go into less than 1 or greater than 1 as a branching ratio metric. What was super interesting was when they were put in the game world, they kind of fell back into that branching ratio of 1.
In the game world, once they're in the computer.
Correct. Their branching ratio and their criticality metrics changed.
And that's based on their self-organization.
Correct.
Is it physical? Are they actually moving around? And
yeah, there's some moving around. Well, the ones that we don't constrain into the wells, they actually physically will just pack
Right.
up and move around kind of thing, which is super annoying because then, you have to continuously assay and, change your
And moving
locations.
around.
Yeah. Which is very annoying because you now have two dynamical systems that you have to deal with and you know how difficult those things are. But, going back to this criticality thing, seeing that result actually made us think that potentially criticality might be a very important function of information processing, i.e., if you are out of the critical zone, you don't process information well enough, right? You have less of a chance of survival. So, we started to look at that, and that's internally one of our metrics of measuring how healthy a network is and how ready they're there to process information. But along the way, we've also observed things like the amount of data used to train these systems is significantly less. I think almost 5,000 times less than your traditional reinforcement learning systems.
5,000 times less.
Yeah,
So they're
backpropagation
learning
system.
much, much faster than
Correct.
training.
Exactly. And we also saw this in a paper. We had a NeurIPS poster that was in 22, and that got turned into a paper that's peer reviewed in 25 that came out, where we essentially did the head-to-head comparison. We took the same game that we gave the neurons, the pong game, and we gave it to 3 reinforcement algorithms. So we had DQN, which is your AlphaGo, the granddaddy of all reinforcement learning algorithms. And then we tried 2 newer ones. There was, I think, A2C and PPO, active critic model. And I can't remember what the PPO stands for. It's been a while. And those are more sample efficient algorithms. Yet the biological system still massively outperformed them, right? Because we sample constrained the game. The game was running 100 frames per second. We only gave it 20 minutes of gameplay. So that's 60 times 100 times 20, which is about 1,200 frames of data. The reinforcement learning agents couldn't do anything with the 1,200 frames.
Not enough.
Not enough, exactly. And
So what's happening here? So then you're converting that into these electrical signals.
Yep.
It's effectively training it.
Yep.
So the neurons are self-organizing, trying to minimize entropy
Yes.
as they're getting these inputs.
Correct.
And then what's the next step to actually get them to play?
Yeah, you just let them play. So there is no. The really cool thing is that there is no training or inference. It's all in the same loop. It's an online learning system. So
explain that a bit.
So in your traditional machine learning, right, you would essentially make it, let's say, make it play 1,000 times, all right, and then you would backpropagate the error signal, and then it would try another 1,000 times with the new.
Fit all the weights.
Yeah, fit all the weights, right? The biological system doesn't do that. If you and I were to play the game, we would play 3 or 4 rounds. And as we're playing those 3 or 4 rounds, we're actually fitting the weights at the same time, thereby reducing the amount of training and inference time because it doesn't exist. It's
and it
part of
really
the
is
loop.
happening at the same time.
There isn't a separate training or an inference, which is really important because if you don't have a training and inference and it's continuously learning, you now have an online learning system, which is very important for tasks where the environment or the task itself is dynamic and is continuously changing.
Yeah, I think also when you think about this $1 billion capex on training that's going on at the moment, it's clearly significantly less efficient than what's going on in our own brains.
And the fact that the large language models have to be continuously updated, right? And retrained. So I don't know, if you open up ChatGPT and click on some debug menu, you probably can see when it was last trained on and the weights were frozen at a particular date and time. So
and they don't learn from, you could use the same model 1,000 times. It's not going to learn anything.
Correct.
Unless you store the information separately.
Exactly. Which is, I guess we're sort of dealing with a problem using RAGs and all that stuff, right? Where they're going out into the web and pulling information and digesting it. But in its internal weights, let's say we discovered a new species from an unknown planet, right? And you give it to the LLM, it'd be like, I have no idea what you're talking about. And I can't help you because it just it cannot change its weights to ingest that new information.
Is there a way of training language models that would be more in line with what's going on in our brains? Substantially more efficient as well?
Possibly. But having said that, language models are very powerful at what they do. Because if you think about it, yes, they did take a lot of time, energy, and data to train on, but no human has the same amount of information stored in their brains as an LLM. No human can figure out the high dimensional trajectory and patterns, right? Or if you ask it a prompt for code and it comes up with some really elegant solution that it was able to do it because it had all of that code repository and could see through the patterns. Our brains are just not good at doing that. So I think, the LLMs are actually very good and very powerful as a tool that we have. How we use it is going to be the real question, right? In many cases, I've always held that the LLM is essentially the next evolution of a search engine, right? Because if you think about why, like from a business perspective, why LLMs are so disruptive is that now anyone can actually build a Google. You could just download the weights, run GPT OSS or whatever the new models are on your machine. And now you have something that is equivalent to Google that would be able to answer your questions in natural language, give you responses back. Whereas previously, nobody had the ability to scrape the internet and index it and give you responses, except for maybe Google and Bing. So I think that's a really disruptive model.
Do you think you'd be able to create a computer version of what's going on in here? If you mapped out what each neuron is doing, effectively modeled that in a computer and came up with a different way of building. I mean, these are kind of computers in a way.
Well, I think yes and no. So I think you're probably talking about a recent paper that came out, or no, I think it was a video. The paper came out much earlier of the fruit fly connectome, right? I think it is possible. I'm not sure how they're doing it because I didn't see the details. My understanding is they use a very simple leaky integrated fire for how to actually model the neuron. Because remember, it's one thing to actually come up with a connectome, which is essentially a map that says, point A to point B, which goes to point C. It's another thing to actually model a car traveling from point A to point B to point C, right? What car are you using? How fast is it going? How many gears and what's the road like? There are all these questions, right, of how to actually make it work. So there's a lot of unanswered questions from that side of things. And I think to really accurately model it might be a little bit too difficult, right? Because if you think about most of what's happening in these neurons, there are very deep partial differential equations that have to be solved to actually model for a neuron firing or not. On top of that, each neuron is individually modeled as opposed to, as a matrix multiplication where you can just do a very large number of operations in one very large calculation in a single operation. So I think it's going to be very difficult. The question that becomes is what level of abstraction matters? Right?
Well, that's the thing. I mean, what do you think does matter? We talked about the neurons moving. There's clearly some kind of spatial element. And perhaps even like the neuron moving is kind of like what you're doing when you're back propagating. You're just adjusting weight slightly to get it better.
Possibly.
And then you have the connectome, which is how they're actually connected.
But
and
remember,
then you have, is it gating potentials? Like what else is relevant in these systems?
So yeah, there's like, the gated potentials, right? Like
That's whether it fires or doesn't fire.
Yeah. And if you think about how machine learning algorithms work, all of them have the same nonlinearity function applied to them, right? So that's like a ReLU or tanh or one of those. It's the same for the entire operation. Now imagine if your activation function was different for each neuron. You probably couldn't do a matrix multiply. You'd have to go through a for loop.
And there are different potentials and
yeah, definitely, because we don't have all the same neurons in our head, right? Even in the cortical cloud, we have started to offer a mixture of cortical and hippocampal neurons at different concentrations, and also supplementing it with glia as well. It is, I think, vastly more complex than how we are approaching it from the modeling computer science.
So there's three different cells: the hippocampal, the glial.
Yeah.
I guess the normal neurons.
Yeah.
What are they all used for in your system, or how does adding them change the outputs?
Yeah. So cortical neurons are essentially like processing neurons, right? Like they're like your CPU. You can think of it that way. Hippocampal neurons, the hippocampus is where we store memories, but it also provides us with ability to do spatial processing, right? So if you have a map and you're engaging in trying to figure out where you are, you're using your hippocampal neurons, right? And glia are essentially the supporting cell structures that keep the neurons healthy. So they clean up the waste, they feed them, and they keep them from getting infections and so forth. So they're very important. We used to think the glia was not really that important at all, but it turns out they are actually really important.
Are they immune cells, or is that too simple?
There's some of them are immune cells, but in your brain, you have all of those cells as well. But the really interesting thing is that neurons don't regrow. Every single neuron you have has been with you since your birth, unlike your cheek cells or your skin cells where you shed them every couple of weeks or so. There has been evidence that some of them do replicate in the deep hippocampal structures, but in general, most neurons never grow back. That's why if you get a stroke, it's just like you don't grow back more neurons. There's a lot of really good work happening in the regenerative medicine space right now, right? This is more about broadly, like naturalistically, this is what happens with neurons. They don't grow. The glia are the ones that actually grow. And, when you hear about brain cancers, they're actually the glia that cause the brain cancers, right? You very rarely ever get a brain cancer from a neuron because they just don't grow. They're very stable genomically as well. So yeah, the more this particular cell grows and replicates, the more unstable their gene structure becomes, and they become a cancer candidate, right? That's why a lot of cancerous cells are like colon cancer. Breast cancer is a little bit different because it's mostly like a lot of them are hormonal activated. Skin cancer, bowel cancer, those things, the turnover of cells are really quick. Anyway, besides the point, they help keep the cells alive and functioning well. When you put all of these together, what we see at Cortical Labs is that you get a pretty good mixture to get some learning performance better than just having one type of its own.
Do they kind of live longer as well? I mean, is it hard to keep these things alive and happy?
Yeah, they do live longer. They do also have better spiking activity.
Spiking activity.
Electrical activity, right? So, we measure
Like sharper signals or more consistent.
More active cells kind of thing. So that's what we do. This is essentially the other side of Cortical Labs. We're still a deep tech research heavy company, right? There's still a lot of unanswered questions. The only difference is that we also have a commercial angle now. And a lot of our research work is actually now focused on developing the best technologies for our users. So if we come up with a better mixture of cells that give us better performance, that gets rolled into products for our users. If we discover a better way of stimulating the cells, that gets rolled into our API so that you can just call it with a Python function call.
Let's return back to this device. So where does this sit in that?
So this would sit in the neural chamber, which is right here. You can't really see it, but we have electrodes down the
I'll take a photo so we can add it.
And that clumps the chip, the exterior of the chip, and makes electrical contact. That's where we read and write the activity. So think of it like our Neuralink, essentially.
So that's in there.
Yep. There's a base compute unit at the back and lower back of the unit. It runs a quad-core ARM chip with an FPGA module that we use very heavily to modulate our signals. And
you've had to build this whole thing, haven't you? You've had to convert those direct electrical signals into something that a computer can use and you can put on the internet.
Exactly.
Pretty serious engineering.
It's a lot of engineering, right? We had to do that and also the information back.
And keep the neurons alive.
Correct. And all the biological. This is a brand new system. So, we just took it from our workshop. But there would be a perfusion circuit that's installed here. But you can't really see it, but you can see some elements of the life support system. So we have 3 diastolic pumps. So these are specialized pumps where you can have a closed circuit, but still pump the fluid through. So it's like your IV lines in the hospital, right? Where there's no pump pushing fluid through. And the way it pushes the fluid is by squeezing the tubes. So these are triple diastolic pumps. We also have a gas.
These will push the fluid through the cell at the.
We also have 2 filtration units, right? One for gas and one for feeding and waste. And they would sit in that region that's currently unpopulated.
Yeah.
Yeah, and that's where the pumps would push the fluid through for filtration so we can get the gas into the cells and also removing the waste material, but also exchanging it for glucose. So they're going through a semi-permeable membrane.
What are the inputs and outputs? Glucose, water, oxygen?
So biochemically, its inputs are glucose, electrolytes, growth factors, pH buffers, which is essentially bicarb.
And this is all work that you've had to do, right?
Yeah.
You've had to figure all these things out.
Yeah, we've had to do it. We've had to do all this chromatography and figuring out, what's the concentrations. We had to figure out what filtration systems we had to build in order to maintain the large molecular weight growth factors while allowing the transmission of smaller molecules. Because you really want glucose to equilibrate across the membrane, but you want to trap the large growth factors. So we had to source a filter with 80 kilodaltons as the cutoff point because we didn't want, albumin is about 150 kilodaltons, which is one of the larger proteins. So egg white is mostly albumin. So we have that. And then we also remove ammonia, which is a byproduct of just cellular metabolism. Some CO2 gets generated as well. And the ammonia really is the one that changes the pH. So we have to keep the pH quite in a very narrow range because the thing that kills neurons and kills humans as well is pH changes, right? So you have alkalosis, acidosis, and that messes around with the potassium transport in the cells. And neurons, being very electrically active, rely a lot on potassium as a ion that goes in and out of the membranes. So we have those. So the system takes in gas, it takes in CO2, nitrogen, and normal air. And then we also have inputs and outputs for the media. And then we have a USB-C, A, and a gigabit Ethernet port.
Get the actual data out.
Correct.
Interesting. That's some serious engineering.
We'll see some of them. I think we might have a few of them stripped and you could actually, look at the electronics behind them.
And how have you built the team to handle all of this?
I think we're very blessed to have a great team, very multidisciplinary, very visionary, right, to make this all reality. And I think it's been a challenge, right, trying to assemble a team that can work across disciplines, come across challenges, and have a vision to build something that never quite existed before. So yeah, it's a mixture of going through LinkedIn, it's a mixture of putting job ads out, friend-of-friend referrals kind of stuff. But we have great talent in Australia, very good talent. We just need places for them to go.
I can see this is the kind of project that would attract smart people.
Yeah.
So interesting and so novel.
I think the novelty factor definitely helps.
And so when somebody is playing Doom on the other side of the world through an API, what's going on in here?
Yep.
They're sending in.
So the Doom code is actually available on GitHub if you really want to have a look at it and play around with the system. And also, a bit of a plug, but the Cortical cloud is now online. So if you wish to also run the Doom code, you can just go to cloud.corticallabs.com and click on it and sign up and, clone the repo and run it. But the plug aside, what's happening with Doom is it's quite interesting because Sean, who built it, actually built it as a tandem system. So it's not purely the neurons doing stuff on its own as it's learning. There's actually a reinforcement learning system, I think it's PPO that he's using, that trains in tandem with the neurons, the encoder and decoder bits. So essentially he's also got convolutional neural net that takes the pixel information and converts it into a spike train that the neurons can understand. And it's all open source, so you can have a look at it and you can run that. So the thing that he's built is that bridge, right, where the pixel information is converted into spike trains, electrical signals that we then can pipe in through our system to the neurons, stimulate them, and then read the responses back out that gets converted from spike trains back into WASD spacebar.
Incredible stuff.
Yeah. There's a little bit more machine learning in that one, but the good news is that if you remove the cells, the system stops playing. And so that's our control, right? If you have a pure machine learning system, if you remove the cells, it should still keep playing, right? But we were very cautious as well. When we saw the results, we were like, okay, Sean, you got to run a couple more controls here. So we ran the controls, and yeah, it turns out that if you give it fake spike data, it just won't actually play, which is part of our SDK as well.
This got a lot of commentary on the internet because if some people were writing about it as though these human neuronal cells were waking up in like the world of Doom and immediately confronted by monsters,
right?
Which I imagine would be quite terrifying.
Well, I think it would be, except
not quite what's going on.
It's not quite what it's going on. I mean, like in many cases, you could replace the same Doom game with, I don't know, My Little Pony or whatever, right? But the objective is still move around and, shoot rainbows at the ponies. It will still play the same way because it has no idea what it's actually looking at. And if you think about Doom, a lot of the whole, oh my God, the demons and so forth is a stylistic and art thing that, it's a cultural stuff for us. If you give it to somebody who's never seen that kind of stuff and no cultural reference, they'd be like, okay, this is just some very weird stuff.
Got it. And so I guess from here, I'd be curious to know your thoughts about, consciousness, feeling, emotion. Like we're talking about brain organelles with human cells, human neuron cells. At what level of organization or size do you think those things come in? Is there anything you've learned or any thoughts you have?
It's a very deep philosophical question, and it really depends on your personal beliefs because it's the hard problem of consciousness, right? Like what David Chalmers says. And the thing here is that maybe the only thing that's conscious in this whole system is just yourself. How do I know that you're conscious? How do you know that I'm conscious? We don't, right? The only thing that we truly know that is conscious is ourselves. So I think this is a really hard question to answer. But there are parameters that you probably can look at as proxies for that. The issue here more is that the field of consciousness is even lacking, I think, an agreed upon consensus of like the nomenclature of definition. What is consciousness, right? What is consciousness and how is it different from sentience? Are they the same thing? But if they are, why do you have two different words to describe it?
Okay, give it to us, Hon. What's your definition?
So
how would you separate them?
I think this is, I subscribe to the paper that, came out in 2022, which Carl defined sentience as any system that was able to respond to a stimulus, right? So in most cases, I would think that, maybe
That's a pretty weak definition.
yeah, but it's like for instance, a paramecium or a single cell organism, right? Can respond to its stimulus, right? You poke it, it'll go the other way. You put some food there, it'll come to the food. But it's only got one cell.
They do look alive when you see them swimming around,
Yeah.
attacking each other,
Exactly.
running away from each other.
Are they conscious? I don't think they are. I think it'd be very hard to ascribe levels of consciousness to that. So I think that is sentient. I don't think it's conscious. And then you have this whole order of organisms that you then work your way through. A very interesting one. So even within species or within the different species, we have levels of consciousness that we ascribe to different species based on our cultural norms. For instance, no one would eat a dog because they think it's more conscious. But if you think about it, a dog is actually as intelligent or as conscious as probably a pig is. Yet we're very happy to eat a pig because we think it's a lower life form. So I think it's one of those, it's a very deep philosophical, maybe even a religious question about what consciousness is. And with what we have with the system, I doubt that it is, because it's really 200,000 neurons with no structure. So you can think of it like a flattened cockroach.
If we talked about parameters, it could be a higher parameter system, right? You've got the geographical alignment, connectome. It'd be higher order than 800,000.
Well, maybe it's also the fact that you might need cortical columns, right? Like neocortex may be involved in
like three-dimensional. Is this a one-dimensional?
Yeah, it's
two-dimensional.
Well, it's 2.5 dimensional, right? Because if you look down a microscope and you'll see it later, we do have like undulations. So you have some areas of high density, low density, but they're not spherical like, the organoids or spheroids as they call them.
Yeah, maybe you go to the limit. Like, is one neuron conscious? Almost certainly not.
Probably not.
Two? Almost certainly not.
And
so what's the current thinking on where that higher order thinking
comes from? I don't think anybody really has the answer to that question. Primarily also because we just don't have the tools to really delve and look into this, right? And if anything that we do know comes from knockout models, right? So mostly from neurology or psychiatry, where we see cases of people who, like one of the best examples, brain damage, right? So here's a really cool one. It's a very well-known phenomenon of people with blindsidedness, where they are clinically blind, right? You put a Snellen chart in front of them, they kind of read a single thing. Yet, if they're walking and you put an obstacle around them, they will avoid it. Or if you throw a ball at them, they will catch it. But they have no conscious experience of catching the ball or going around the chair, right? That's a really interesting, I think, difference, right? So if a person has blindsidedness, they are sentient because they can catch the ball. But if you ask them, do you catch the ball? They'll be like, no, what are you talking about? Or why I'm holding a ball right now. I have no idea how I got this. So there is no conscious experience of that happening. So you see, this is the reason why we don't really know much about it. And all we can see and all we know from these two systems and definitions just from watching people with neurological defects.
Yeah, I heard there were people who were blind who could almost tell what was going on in a room by the sound.
Yeah.
Almost like proto-sonar.
Well,
if you're talking against a wall, you'd probably be able to tell without that sense being heightened.
I think that it's very well known fact that people who are born with low or no vision actually route a lot of the auditory signals into the visual cortex. So they actually see sounds, and I guess bats doing sonar. And there are people out there who have actually been able to navigate spaces just by listening to the echoes. Yeah.
Heard that. I think it's an evolutionary thing. It's one of those things, well, how could a bat learn that? But if people can almost do it, it's much more straightforward to see the evolutionary.
I think it's also just the power of the neuroplasticity of the brain, right? A bat has eyes, it can see things. But if you're flying in the dark and you get no photons, why route all that information to the visual system when you can get better information from acoustics, right? So I think that's the other bit of the equation, which is the brain is always seeking information.
So you've worked with systems with 800,000 neurons. How many neurons are there in the brain?
Probably 54, 60 billion neurons, I think.
Do you think if you had that many neurons in one of these systems?
Oh, sorry. Actually, no, I think it's a billion, but it's probably 60 billion synapses. 60
billion synapses.
Yeah.
I'm just trying to figure out what happened if you scaled this up. Would you get a system that could do more than play Doom?
Maybe. Who knows? I mean, it's one of those things why we built the CR1 as it is in the cloud component, right? Which is coming as a feature, ability to actually network multiple CR1s together to
Multiple of these things together.
correct, yeah, because we have Ethernet ports, so we can probably shuffle information through UDP packets from one system to the next and come up with pseudo structures, right? So this is the next phase in some of our research work. Can we do this? And then once we work on this and we have a solid framework, we'll release it as an API update, and people will be able to start scaling up and stacking multiple CR1 units together for more processing, maybe multiplayer games. I don't know.
You've got these neurons too. So to play Doom, they're doing what? 4 directions and a fire button? Is it so 5?
Pretty much, yes. Yeah.
Could you give it, say, mid-20s outputs and get it to output text?
Well, I think
speak back.
I think somebody is doing that already. So one of early access preview customers hooked an LLM to the same person, also hooked it up to a crypto trading bot. And I think you're losing money if you use that. But it did make some interesting trades based on historical data.
Yeah, well, I guess in this instance you would put in that text stream.
Yeah. But then again,
and then kind of like a large language model, maybe it would start learning what are words, what makes sense.
Who knows, man? There was like
speaking back.
Wasn't there a video of a guy who made a goldfish bet against Wall Street bets, and the goldfish made more money than Wall Street bets, I think. But then again, they're all a bunch of apes and regards. So
yeah, I feel like this is different. I feel like if you've got like these like hundreds of thousands of neurons together,
yeah,
and they start saying words back, it's a bit different to like, I don't know, an octopus World Cup winner.
Maybe. Who knows, right?
You're right.
This is your chance to experiment, right? This is where having a platform with minimal amount of setup required is just going to be very useful. These questions will no longer be wouldn't it be cool, if you could do this in the pub kind of question, but like, wouldn't it be kind of cool? I'm going to try this out tonight. Maybe I'll burn like, $2,000 on this for a month. But hey, if it works out, you've come across something really interesting.
And I guess that's a key part of what you're doing, right? This is
Correct.
a business. So what's the model?
The model really is, infrastructure. And if you think about the whole AI space at the moment, the AI players are free to do what they do best, which is research and building like software, because they don't have to worry about hardware. That's taken care of people like NVIDIA and TSMC. This field has no TSMC or NVIDIA, and you really have to start with those guys. So that's where we're hoping to fill the blanks, right? And help our customers and our research partners go faster. And it's a question as well that we've had even from investors, which is, if you have such great tech, why do you give it away? Why do you open it to people to use? And the answer I always have is, if you look at historically how the AI space came about, it's really because of AlexNet. And for the longest time, neural nets were just so underpowered. Nobody knew how to make them work until Alex Krzyzewski was like, hey, you know what? I have a GPU and I learned a little bit of CUDA. Maybe I should just build a convolutional neural net using CUDA. Does that. Overnight, he, beats ImageNet by 8 percentage points and changes the field forever. This kind of thing, nobody could foresee and predict, right? So I think, it's hubristic for anyone to say, I have the solution to this. And really, we need to think about working collaboratively as a community, exchange ideas, propagate it through, and maybe the least expected person might actually come up with the solution.
And this people can access this now. So if somebody has an experiment or an idea they want to run, and what's the commercial model? Are you going to sell these? Are you going to just rent them out on the cloud?
We do all of the above, right? So, we sell these for about $35,000 to research labs. So we've had at least 8 units go out in the last quarter. And those are, highly established like laboratories like in Dartmouth or Tokyo University and University of Bologna that have the ability to culture cells. And they have very deep questions that they want answered using this technology. The converse and flip side is, if you're not a biology lab, but you still want to get involved with this, the cloud model makes the best sense.
Yeah. I guess if you bought one of these, you'd still need all the systems around it to keep the cells alive.
Yeah, and you need to be able to grow neurons and keep them alive. And
why do you use human neurons, by the way? Why don't you use... I think that's materially different to using a rat or something.
We have chimp neurons as well. But the reason why we use human neurons is because there's a lot more literature behind it. There's a lot more funding that has gone into the space from the biomedical research angle. And as in most science, we're standing on the shoulders of giants, right? And so I would love, and Brett, my CSO, would love to do cephalopod-like neurons, the whole octopus Cthulhu kind of thing is a bit of an obsession. But nobody really sees any value in harvesting octopus neurons or growing them stem cell wise, because there's no commercial output. It's just curiosity. So unless one of your viewers here is a billionaire and is willing to fund it, it's going to take a while before we get there. On the flip side, there's a lot of clinical trials. There's a lot of funding that goes into like disease modeling and so forth in the human space that, we're able to just take the corpus of knowledge and go quicker with it.
And contribute to it.
Exactly. Yes.
Fascinating. So what's next? Is there going to be another version of this?
Yeah.
Is there a product roadmap
that
you're able to share?
Well, yeah, we'll probably go with the TikTok model as well. Are you familiar with that?
You'll have to explain it.
Okay. So in the hardware space, there's a common development cycle known as the tick and the tock. Intel used to do it. Actually, all the silicon guys do it. Even Apple does it, right? So what was it? You used to have like an iPhone 4 and an iPhone S and then a 5 and an S or something like that. I think they abandoned that model. It's still mostly the same, but the naming scheme is different now. But the TikTok model essentially says every second version, there's a major revision, right? But the interim, the talk to the tick, is a incremental development on top of that. And that is a way of spreading out development costs, getting a recoup on the ROI and so forth. And so for us, we're going down with this TikTok model. This is the tick. The next talk is going to be a CR1 Pro. We'll give people more electrodes to play with. We'll give them more processing power to handle, larger data sets and so forth. Really like going from a MacBook Air to MacBook Pro kind of thing. The CL2 is probably coming in the next 48 months or so. We've started to think about ideas. I can't really reveal too much of it, but expect like miniaturization of this stuff, new modalities of interfacing with cells, new modalities of packaging cells and stuff like that. So it's all very exciting stuff, but we need to do a bit more R&D to firm up on how that's going to look like.
Is there any interesting things about neurons that people are unaware of?
Interesting thing about neurons that people don't know. Well, I guess this is a relatively recent discovery. People, back to your question of why do we use human neurons instead of mouse neurons? Previously, we used to think that all mammalian neurons are the same until I think about 3 or 4 years ago, a group at MIT actually did a very interesting study where they harvested a whole bunch of like throwaway temporal lobes from biopsies and patch clamped the neurons, the human neurons, and they noticed that
human patch clamped.
So yeah, patch clamping essentially is just putting 2 electrodes on a neuron, firing an electrical signal, and then like watching it propagate through. You get a lot of information from doing that, but it's very painstaking and painful process for the operator because it's very finicky work. Anyway, they patch clamped a whole bunch of neurons and they found that the human neurons on average is usually longer and bigger than a mouse neuron, but they have the same number of ion channels. So what they think is that humans are probably better at information processing because it's just bigger and you can hold more electrical states without reducing the fidelity as you would have in a mouse.
What do you mean by hold more electrical?
So if you have more electrical...
different parts of the neuron have different charges effectively?
Essentially, yep.
And is that where the other neurons are connecting? Is that...
No, it's just because if you think about like how an electric potential works, it's like a Mexican wave, right? And, physiology 101, electric potential or electrical spike of how a neuron sends a signal from its body to all the way to the other end of the axon happens as a threshold function. So enough excitation happens and the ion channels open and you get this propagation of the signal through. But it's all highly coordinated. And if you have more electrical states per se in the synapses, which is where the terminus is, and also on the axonal body, you could have more degrees of dimensionality that you can hold that information before it actually fires or to fire.
Because it comes back to that point around what are the parameters of these networks. It could be much higher.
Yeah, much higher. Much higher. Because also remember the other thing, so it's a very interesting thing about neurons is that because they're not doing a back pass, right, a back propagation pass, it's probably one of the reasons why it's so efficient, right? Because if you think about your traditional GPUs and you're doing like a back pass, one of the reasons, and this is actually a really interesting like physics phenomenon, an information theory one, that a GPU heats up so much is this thing called Landauer's principle, which is that the energy expanded mostly when you're doing a compute is in the erasing of a bit. And if you actually, and this is actually a very interesting one, like, I think we were talking offline about photonic computing and, a host of all these exotic computes. There's also another space called reversible computing, where if you can do your operations without actually erasing a bit, you actually don't produce much heat and you're very much more efficient. And so
Explain that a bit. Explain that again. So you do an operation without flipping a bit,
without erasing,
raising.
right? Because if you think in, let's say like you have a sequence, like an array of like 0, 1, 1, 0, right? If I wanted to change the sequence to 0, 1, 1, 1, rather than erasing that last bit, if I just added 1 to that, you now have the same output 0, 1, 1, 1, 1, but you didn't erase a bit and fill it in with 1. So the operation of +1 gives you like almost no energy like loss or expenditure. On the other hand, if you were to just really dumbly just go, oh, I'm just going to erase everything 0, 1, 1, 1 to like nothing and then fill in the numbers, you're expending energy on top of that. And if you just erase the last number and you fill it with the next number, then you've also expended energy. So it's a very interesting space of reversible computing. It has some like spillovers into quantum computing, but information theoretically, that is the reason why, you know, the current architectures generate so much heat and are so inefficient, right? Because every time you do a back pass, you're actually erasing the bits and you're really filling them again. So biological systems probably are doing some sort of reversible computing where we're not erasing bits, and that's why we don't.
Yeah, I guess as you're doing something or seeing something, those networks are organizing.
Yeah, exactly.
Live.
Yeah.
What's your take on quantum computing? What?
My take, I don't know. I think it's overly valued, right, as a space, because
I think there's a financial question as well.
Yeah, it's attracted so much, but for so long, what have we seen from it? I would have thought that, you know, from the trillions that have been raised, we would have something workable by now. But, you know, all kidding aside, I mean, the thing I think that quantum computers are going to be really good for is, as Scott Aronson says, it's quantum simulations. There's a lot of conjecture about, is it going to be faster than classical stuff and so forth. And I think the only thing that's proven to be faster than classical computers is Shor's algorithm, right, which is factorization. But I think we only managed to factorize up to 21 or something like that because we just run out of
don't have enough of
qubits, right? Theoretically, like plausible. Problem is, how do you actually build it physically, right? And I think that's where all the money's going at the moment.
Yeah, I get asked that a lot. I try and say, look, it's still at the maths phase. Like, they don't even have the algorithm, the actual algorithms to use to do all the things that are being promised to the financial speculators.
100%. And I think this is, you know, the same problem that we have in neuromorphic space, right? Where you have like a whole bunch of these neuromorphic systems, but what is the algorithm that you're going to use to train these things that will give you the best yield and output? And I think, you know, for us, we hopefully try to sidestep that question by saying, well, let's just rely on the neurons to do that heavy lifting and then
do the organization, do all the,
which is
whatever it is they're doing.
I know it's a really bad cop-out answer, right? Because as a scientist, you're like, that's not the answer. You know, it's like asking, why is the sky blue? Oh, well, that it just is. No, you need to know because of the angle of the earth and the scattering and all that stuff. That's the correct answer. Right now, the answer that we have is the same thing. You know, why is it doing it like this? Or it's just doing it because that is how it is.
That's why this is so fascinating, because you're doing engineering with it and manipulating and working with the system.
Yep.
There's like a certain kind of knowledge that comes from doing that.
100%. Exactly.
That you wouldn't get otherwise.
And I think this is going to go the same way as, you know, neural nets, right? The fundamental, we don't understand how neural nets work. We get all these answers. And, you know, that's why we have all these questions like, why are they hallucinating and so forth? Because we just don't have a mechanistic way of proving or coming out with a solution of why something is happening in neural net space. But we've made so much progress because we have the tools and people can engineer and build things. It's like Romans building bridges without understanding Newtonian physics, right? You build enough bridges, eventually you come across a formula that guarantees it doesn't fall down without understanding forces and gravity.
Well, brains are the best example of that, right?
Exactly.
You're not doing calculus when you catch a ball.
Yeah.
You're just calculating it somehow.
Yeah.
And putting your hand in the exact spot.
Exactly. Well, if you think that's cool, I should send you the video on, and you can show your viewers as well, a dragonfly. So dragonflies are the perfect example of an organism that is predicting ahead of time, because you can see as they're in the video, as an object is falling, it's actually not chasing the object. It's actually coming up with the interception point, and it's flying in the interception point to get the falling object. And if you think about it, that's essentially predicting ahead of time. They're not doing some calculus. Oh yes, if I fly in this trajectory and it's flying this velocity, I'll hit it at this point.
But on some level, they must be doing that.
Must
On
be.
some levels, that calculation must be going on.
Exactly. And only with so few neurons, that I think is incredible.
A lot to learn.
Yes, indeed.
Okay,
Well,
let's
learn with
check it
us,
out.
hopefully.
Absolutely.
All right, let's go.
Some of these you can turn on online.
What are the numbers on them?
Temperature, so it's 37 degrees, carbon dioxide 5%, oxygen 19.7. So these ones do not have any nitrogen pumped into them. So that's why they're at 19%, which is, partial pressure of oxygen in room air.
Oh, what about the green ones here?
Green ones are the slightly older edition. So we did a first edition and then had to go through a rebrand. And so this is actually, these were the OG CL1 units. These are essentially the revision B that we are shipping to customers now. The circuits that we are doing.
So these are the circuits with the...
Yeah. Oh yeah, here we go. That's a chip with the neural activity being recorded. So every time there's a spike or actual potential, we flash the background with some color change, and then we mark where the neurons are.
There's a live experiment going on now.
So there's no experiment, it's just reading the activity and see how the health of it is. So we also plot out, you can see the actual spike waveforms over time. Yeah.
What does each one represent? That's a point?
That's
an electrical junction?
Yeah, so there was the grid. These are that little black dots, and that's where we sampled the activity from.
Got it.
All right. Cool.